Okay, welcome to Unit 3. Here's where we left off in the last lecture. Um, we had our universal common ancestor to eukaryotes, a couple of groups at the very top, Excavata, the SAR, clade, and the Archaeplastida were all examples of basal eukaryotes. The Archaeplastida actually is the one that gave rise to plants. And coming down from the bottom part, we've got the Uniconta. Uniconta are unusual in the sense of having only a single flagellum. And within the Uniconta, we've got the Amoebozoa and the Opisthoconta. Opisthoconta are the ones that are unique in having the flagella attaching from the rear. And animals are within the Opisthoconta. Also in that group are fungi. And there's a third grouping here, which are the choanoflagellates. This is basically, as you can see, our sister taxon. I mean, as animal sister taxon, according to this tree, would not be the fungi. Choanoflagellates are more closely related to animals than are the fungi. And so really a good place for us to begin this discussion would be to talk about that particular group of protistans. They're not animals, they're not fungi, they are outside of the animal kingdom, but they have a lot of characteristics that could point us in a direction towards understanding the origin of our own kingdom. All right, so let's take a little bit of a look at the choanoflagellates. Choanoflagellates comes from two words. The flagellate part is obviously the fact that they have flagella. Choano refers to a collar, like the collar on your shirt. And the way these things are shaped is basically they've got a flagellum and the flagellum extends out from the middle of a collar. Basically it kind of looks like this. Right, so here's the cell. You've got this collar and the flagellum is basically attaching to the end of the cell. Right, so you've got a flagellum, single flagellum, it attaches from the rear. The flagellum pushes water from, this case would be pushing water towards the right, which would cause the choanoflagellate to be propelled in the direction to the left. Okay, so this is a weird little unicellular organism, and it's got a couple of really interesting properties. First of all, the collar is kind of an interesting feature. What does it really do? Well, maybe it reduces the turbulence wake. I think the most important feature of that collar is that it provides an opportunity for the animal, not the animal, for the cell to feed as it swims around. Basically, the water, let me change the color over to blue. Water is pushed out of the collar by the waving of the flagellum. And so the water has got to be taken in from the very base. And so at the very bottom of the collar, you've got little holes, which are like a little sieve that allows water to flow into the base of the collar. Basically, it's pushed in this direction. It comes out of the collar and propels the cell forward. Okay. Now, as it's happening, the cell is moving forward, of course, and the water is moving across the surface. Water is basically drawn into the base of the collar and it gets pushed out from the larger end of the collar by the action of the flagella. Now, as this is happening, the animal is also feeding because the food particles, I'll do that in kind of like an orange color, the food particles, which are suspended solids, are there in the water and they basically pile up at the bottom of the collar because that's kind of like a little filter. It's kind of like the lint filter in your dryer. And because you've got such a large quantity of this organic stuff, the food of the choanoflagellates, they can then take in that food by phagocytosis. And so if we go back to a brownish color, you can sort of say, okay, well, the cell is going to be forming these phagocytic vacuoles. This is actually an indentation towards the middle portion of the cell, and the food particles, the suspended particles, are taken into that phagocytic vacuole, and eventually you end up with a phagocytic vacuole with a whole bunch of organic stuff inside of the cell. Right? And then you could have digestion taking place. And so the cell's digestion is occurring intracellularly. This is a food vacuole. And that's basically how the animal, not animal, that's how the cell eats. It moves and it feeds using the same mechanism. This is just one example of filter feeding. Okay, so that's a little bit more detail than you were probably anticipating getting about choanoflagellates, which is generally a pretty obscure group of protistans. Um, but this becomes the starting point for our investigation of this bigger question, this question that we had up here. The question, like right along the lineage that leads from the common ancestor. Now, if this guy right here is a common ancestor shared by choanoflagellates in animals, what happened there that gave rise to animals? Well, I guess one thing that you could identify is the evolution of multicellularity. Choanoflagellates are clearly not multicellular, and animals clearly are. And so, what was going on there? Well, it turns out that there's some interesting choanoflagellates that are not entirely kind of like the one that I drew above. There are a lot of choanoflagellates that are colonial and still unicellular. And what these guys do is they will take up residence on maybe a rock. This will be a rock. It's a substrate. And the cells, instead of swimming around freely in the area up here, kind of like the ones that we were drawing before, maybe this would be a free-living choanoflagellate, um, there are choanoflagellates that will take up residence by basically pointing themselves kind of nose down on a rock, and they start to filter feed kind of as a sessile organism. They become a sessile unicellular organism and they filter feed. They take in food from the suspended solids that are in the water surrounding this. Eventually, they'll grow up, they'll split, and they start to occupy a little area in which all these clones from the choanoflagellates are attached to each other in what ultimately becomes a kind of a branching structure. You could have a branching structure that splits off again and again. And at the tips of these branches, you'll have these choanoflagellates. They're all descended from the same clone. Basically, one cell, ancestral choanoflagellate cell, is going to give rise to several descendant cells that are all attached to each other, and they are forming like a little tree. Okay? It's a weird thing to think about here because we've got something that looks more plant-like than animal-like. But really, this is the same organism as the one we started off talking about. Our choanoflagellate is unicellular because there's no real differentiation that occurs between the cells, and they're colonial. And I guess the question is, well, what makes this guy not multicellular? And the answer, of course, we've done this before, is that a multicellular organism has got to have differentiated cells. Some of the cells have got to be doing some things, other cells will be doing something else. And because all the cells in this unicellular colonial choanoflagellate are pretty much doing the same thing, this is clearly not an example of true multicellularity. All the cells are behaving the same way. They're all taking food particles in and they're digesting that material intracellularly. Okay. Now, this is actually kind of an interesting starting point because we have today unicellular, free-living unicellular choanoflagellates like the one that we drew up here. We've got some colonial ones like the ones we have down here. And we're going to be skipping forward a little bit and talking about the general structure of a sponge because in our most basal groups of metazoa, most basal of metazoa, that means the most basal of the animals, a sponge is a structure that's got a shape that pretty much ultimately kind of looks like this. Right? It is multicellular. There are many kinds of cell types. When you look at the sponge on the outside, now there are several very complex sponges like brain corals that are very large and they've got a lot of holes in them, but the most basic shapes of the sponge will pretty much just look like a little blob of cells with a little hole at the top. Okay, it'll kind of look like this if you saw it from the outside. They also have lots of pores on the outside, and this is where we get the name porifera. Porifera is the phylum. that sponges belong to. So porifera, if you cut this one in cross section, if we were just to do kind of like a slice right down the middle, you'd end up with a structure that kind of looks like this. At the very top, you've got a hole. This hole right there is the same as this hole right there, and that's called the osculum. Okay, osculum is the big hole that's at the very top. Osculum is where the water leaves. Now you've got these little holes around the outside that are called the ostia, ostium singular, ostia plural. So basically I put little holes in here, and these ostia connect to little flagellated chambers that open up to the inner space of the sponge. Right? Okay, so these are the flagellated chambers, and this empty space in the middle is called the spongocoel. When you see the root word C-O-E-L, that's pronounced seal, just like the animal that balances a ball on its nose. Okay. In the general flow of water through our sponge is as follows. Okay, water from the outside, which carries its load of suspended solids, it enters the sponge through the ostia, and it flows into the flagellated chamber, and then from the flagellated chamber it's pushed into the spongocoel. And then the pressure of all the water that's flowing into the spongocoel from all these flagellated chambers is going to generate positive pressure, which is greater than the pressure that's outside of the osculum. So we'll have bulk flow of water from the spongocoel to the outside world. Okay. Now we're going to draw this again. We're going to draw this as a in kind of like a larger version. I'm going to take this portion right here, and I'm going to blow it up. So on the outside, we're going to have the outer skin of the sponge, and here is the opening to the canal, and here's the flagellated chamber. Here's more cells on the outside, and the flagellated chamber is going to connect to the spongocoel. Okay. Okay, so I don't know if you could see this. The spongocoel is on this side. The outside is on this side. The ostium is over here. The flagellated chamber is the part in the middle. Now's the part where we get a chance to talk about the different kinds of cell types that are there in the sponge. We've got the outer layer. Outer layer goes all the way around the outside of the sponge is the pinacoderm. We also have pinacoderm on the inside surface of the sponge. The cells, specialized cells that control the opening and closing of the ostia are called porocytes. And then we have cells that are lining the flagellated chamber, the ones that actually have the flagellum. And these cells are called, wait for it, choanocytes. Why are they called choanocytes? Well, it's because they've got collars and flagella that beat the water and they basically push the water from the from the flagellated chambers to the outside world. We've got more sponges. We've got more choanocytes over here. They're pushing the water in this direction, which creates that positive pressure inside of the spongocoel. Okay. Okay, so we've got 1, 2, 3 kinds of cells. There's also one more kind of cell, which are called the amoebocytes. And amoebocytes are there inside the general space. And amoebocytes look a whole lot like amoebas. Sometimes they're called archaeocytes too. But what they do is they communicate, they transport the nutrition that's gained from the choanocytes to the rest of the sponge. Basically, the only part of the cell that's actively taking up nutrients are the choanocytes, because these are the cells where you have intracellular digestion, and these choanocytes tend to be very well nourished. They've got extra food. They can pass that food on to the rest of the sponge, but they have to use some type of mechanism to get that food to the rest of the cells. And the amoebocytes are kind of like the Uber Eats cells of the sponge body. Okay. The reason why we're doing all this detailed anatomy, we're not going to do this for every kind of animal. The reason why we're doing this detailed anatomy for the sponge is because we've got this smoking gun. We've got these choanocytes here. that look enticingly like these quantiflagellates that we have that are just outside of the animal kingdom. Remember, coming back up here, if you look outside of the animals, who are the most basally animals right here? Those would be the sponges. Who are the closest relatives to the animals? Well, those are the quantiflagellates. And lo and behold, we actually have a cell type that's shared between sponges and the quantiflagellates. Okay. So that's kind of like a general standards issue story that goes along with the origin of animals. The difference of sponges, of course, is that we've got multiple cell types. We've got at least 4 cell types, and sometimes we've got other kinds of cells as well. Sponges are definitely more complex, but the origin of multicellularity is probably something that happened right along this juncture. What actually happened between point 1 and point 2, that would be the origin of multicellularity. Presumably some type of ancestor that did not have multicellularity became truly differentiated at that point. Okay. And so now I'm going to play this little video clip in which this dude is going to be squirting some fluorescent non-toxic dye close to the base of these sponges. All right. And so you're going to be seeing the dye being soaked up right through the pinacoderm. They're going to go in through the ostia into the flagellated chambers. And the flagellated chambers, you don't see this part, are going to be pushing the seawater and the fluorescent dye into the large spongiocele, that big cavity in the center of the sponge. And the fluorescent dye, as you can see, is coming up right out through the oscula, like little chimneys. All right, so there you have it. Circulation in a sponge. Okay, so now back to this tree. So we're at a juncture where there's strong evidence supporting the idea that there's a single origin for animals. There's a universal metazoan, that means an animal ancestor, which is the progenitor for all animal life as we know it. This is an animal that would have occurred in the Precambrian, sometime probably deeper than 800 million years ago. And it's not until we get to the Phanerozoic where we've got a really good depiction of what happens after that. Here, let me recenter this a little bit so you get to put this in an appropriate location. And so, yeah, so there are actually a few taxa that are of interest when we think about the origin of the animals. One of them are the sponges. Then you have another group called the cnidarians, which includes sea anemones, jellies, box jellies, corals. Then we've got a couple of interesting groups. One are the ctenophores, which are the comb jellies, and then there's of course the bilateria. Bilateria means the bilaterally symmetrical animals from which we eventually get the majority of the animal kingdom that we care about, like fishes and other vertebrates, as well as the arthropods and the mollusks and the worms and that kinds of things. So we've got these 4 different groups that are all descending from that universal common ancestor. We know that there's a relationship there. These are all descendants. The question though is, what are the relationships between these early lineages that branched off from the universal metazoan ancestor? Who came off first? Who came off seconds? Who's the most basal of all the animals? And so there's a lot of discussion about that. There's a perspective taken by the textbook, and we'll be going primarily with that one, but I should probably advise you that there's a lot more discussion about that particular issue than you'll probably want to care about. So just quickly, let me present to you a couple of the competing ideas. So in the traditional picture, and this one is probably closest to the textbook, porifera, the sponges, is the most basal of the grouping. And ctenophores, cnidaria, and the bilateria, they come off as a sister taxon to the porifera. This is probably the traditional picture that you might hear about. Another idea is that there might be some sponges that are more basal and other sponges that are actually sister taxa to the cnidaria, ctenophores, and bilateria. So if you look at sponge paraphyly, that would mean that there are some types of sponges, these guys in particular, for whom you are a closer relative relative to these sponges that are down here. Basically, we've got the S group of sponges and the C group of sponges, and these C groups of sponges are more closely related to you and jellyfish than they are to this guy. Why is that? Well, because you share a more recent common ancestor, right? Another picture might have the ctenophores and the sponges be sister taxa, and the cnidaria and the bilateria being the other group. And there's even a perspective that has the ctenophores as being basal. So that's really where we are in this problem of what happens at the base of the animal tree, but we're not going to worry about that too much. We're going to come back to here and we'll be recreating this tree. We'll be recreating the topology here, focusing on the relationships that you guys have in your textbook. And so the relationships here, as we're going to draw it, is that we'll have a basal grouping leading to the sponges, which have their own diversity of different kinds of sponges. This is all the phylum Porifera. I gave you that word before. And the other grouping is the grouping that will be giving rise to the majority of other animal taxa. In fact, this group is sometimes referred to as the eumetazoa, meaning true metazoa. The sponges, the poriferans, are sometimes referred to as the parazoa, meaning to the side of animals. And then the eumetazoa are going to be giving rise to two major taxa, one of which is going to be the radiata, of which one particular lineage of great importance is going to be the phylum Cnidaria. The other lineage, great importance, is going to be the Bilateria. And the Bilateria is the group that includes all bilateral animals. It'll be splitting this one into two larger domains, one of which is going to be the Deuterostomia. This is a group that will include ultimately two major taxa for us. One of these taxa is going to be the echinoderms, which are the sea stars and sea urchins and the like. Then you've got this other group, which is the group that we tend to care about a lot because it's our group, the chordata. Okay. And so that's the Deuterostomia. The other major group of the Bilateria is going to be the Protostomia, which is a major super kingdom of organisms, which is itself broken into two super kingdoms. One major lineage is the Lophotrochozoa, which is going to include things like the annelids, mollusks, as well as flatworms. It's a platyhelminthes. And the other group of the protostomes is going to be the Ecdysozoa, which is the group that includes the probably arguably the two most successful animal phyla in the whole animal kingdom, which are the Arthropoda, Arthropoda if you prefer. That's insects, crustaceans, spiders, as well as the Nematoda, which are the roundworms. Okay. So in effect, we've got one Ecdysozoa, two Lophotrochozoa, three Deuterostomia, four Radiata, and five Parazoa. Five major clades of kingdoms occurring within the metazoa, within the animals. And so now if we go back to this universal metazoan common ancestor, you can ask, well, what are the characteristics that separated them from the common ancestor that is shared with the choanoflagellates? And one of them, of course, would be multicellularity. If you look at these five super kingdoms of animals, one of the things that they share in common that distinguishes them from the choanoflagellates would be the phenomenon of multicellularity. You call it a synapomorphy of the metazoa. What might be some other synapomorphies? Well, one of the things that has to go along with being multicellular is that you have to direct the development of the organism from a single-celled zygote into a structure that's going to have a particular shape. You're going to have to set up the rules for differentiating the cells and to give rise to the multicellular organism. And this is where we get into some really basic and truly old school lessons in biology. Things that would have been on the table from 100 years ago, maybe even 150 years ago, because we'd been studying embryos and how they develop and how we actually end up with a multicellular organism. It's something that's been fascinating us for a really long time. And so the next little bit of information is as true today as it was in the 1850s when people like von Baer were studying the development of embryos, even before the time that Darwin had published his Origin of Species. So the earliest stages in development belong to a group of cell divisions we generally refer to as the cleavage divisions of embryogenesis. And these are the cell divisions that start off with a zygote, which is simply a fertilized egg, and ultimately it gives rise to a multicellular structure in a cluster of cells that might not actually have the characteristics of true multicellularity, but it's right at the juncture that will ultimately lead to the next bit of development, which will lead to true multicellularity. So think zygote all the way to generic cluster of cells, which is probably undifferentiated if you looked at it, which would look generally pretty undifferentiated seeing it from the outside. There might be some stuff going on inside of the nuclei, but from all appearances, it looks like a completely undifferentiated set of cells, and it might look like this. Yeah, think raspberry. And so in order to get from point A, the zygote, to point B, the cluster of cells, we have to undergo some simple cell divisions. And these cell divisions take place pretty much in rapid fire sequence. You have to duplicate the genome, but we don't have lengthy G1s and G2s in the intervening interphases. So we undergo mitotic cell divisions. The first plane of cell division is going to be going like right from the top to the bottom, and that's going to create a two-cell stage with a cell on the left, a cell on the right. And the next cell division is going to be in the plane of the board or your sheet of paper. So if the first cell division splits you right and left on your screen, this one is going to split the right-left embryo into a right front, right back, left back. Visualize it this way, leaving you with an embryo that looks more or less kind of like a bell pepper. The third plane of cell division is going to be horizontal. So this is going to separate the top tier of cells from the lower tier of cells. And here's where we end up with a cluster of cells with four on top and four on the bottom, looking more or less like that. And from this point forward, obviously, you're not going to be able to identify particular planes of cell division, but we'll continue to have more or less synchronous cell divisions going on until we end up with this generic cluster of cells. And those are the cleavage divisions. Okay. Now, following the cleavage divisions, we have a period of true morphogenesis. True morphogenesis means the origin of shape. And the operative term here is a word that we'll be identifying as gastrulation, which is basically the formation of an embryonic stage called the gastrula. And so, at least in some embryos, we have a situation where this initial cluster of cells we have right here, okay, here is the thing we'll call the blastula. At the very bottom of the blastula, like right around here, you'll have what's traditionally been called the blastopore origin. We'll form the blastopore there, which is a hole. And all the cells from all around the outside of the embryo will start migrating toward the blastopore, and from the blastopore, you're going to see them diving towards the middle, which is going to create an inside layer. So you'll have these cells that are going to come towards the middle, creating an inside layer to the blastula. And on the outside, you'll be left with the cells that have kind of migrated, but they stayed on the outside of the embryo. And ultimately, what this is going to do is it'll give rise to an embryo that's got a complete inner layer and an outer layer, and that's going to be the gastrula. And the gastrula has got three layers. If we sort of visualize this, you'll be a layer of, those would be the cells that surround the outside of the embryo. And on the inside of the embryo, you'll have a layer of cells that are the cells that were able to make it to the blastopore, dive inside to create this inner layer. And so now we can go ahead and put some names on this. The whole embryo at this stage, you might call it a gastrula. And the two layers that we have on the gastrula, one on the outside we'll call the ectoderm, and the one on the inside, naturally, we'll call the endoderm. The blastopore is still a structure that you can see on the outside of the gastrula, and it's the opening that goes into the inside space. The inside space in the middle is referred to as the archenteron, which literally means the ancient gut space. That's the part that will eventually become the stomach of the animal. Okay. Now, if you were to actually see the gastrula from the outside, in other words, not a cross-section like what I've drawn there, it would probably look more or less like a little globe with a little hole at the bottom. Okay. The hole at the bottom is the blastopore, be P. And if you were to walk through that blastopore, you would end up in a large space, the archenteron. And from that large space, you could feel in all directions, up, down, right and left. You could probably feel, apart from the blastopore, you would be touching endoderm. And then if you were to drill through the endoderm, you would have a little space before you reach the ectoderm. And if you drilled through the ectoderm, you'd be back on the outside of the embryo. So how does this relate to our earlier question about what were the other synapomorphies of the metazoa, of the animals? Well, it's funny you should ask that, because what we're looking at right here with a gastrula embryo is kind of like the starting point of the body plans for some of the simplest animals. Basically, in some animals like Cnidaria, like sea anemone, the basic body plan kind of looks like this. You've got a base, you've got a crown of tentacles. These tentacles have stinging cells, and then you've got a little mouth over here, and there's a large empty space in the middle. For a sea anemone, we can identify the basic parts of the anatomy. For example, this would be the mouth, this would be the gastrovascular cavity, the outside would be the epidermis, and the inside layer, this layer on the inside, would be the gastrodermis. And what I'm basically saying here is that if you look now at the basic structure of our gastrula embryo, you've got all the parts. You've got a blastopore, which is kind of like the mouth. That's the part that will develop into the mouth. The gastrovascular cavity is going to become the archenteron over here. Ectoderm is going to become the epidermis, as well as the tentacles with all the stinging cells around the mouth. And then you've got the endoderm, which is going to be developing into the specialized structure called the gastrodermis, the thing that will digest food, secrete enzymes, and absorb the food. So yeah, I mean, a sea anemone is basically a gastrula that's been elaborated somewhat. And in the general picture of animal development, you could say that once the animal has reached the gastrula phase, it has become a true multicellular animal. Up to the point where it's just kind of like a generic cluster of cells over here, you might say, well, those cells, they haven't really differentiated from each other. They might at some point reach the point of multicellularity, but right now they're not really there. Once we hit the gastrula, it's simply a matter of taking the existing cells of the gastrula, which have now differentiated into ectoderm and endoderm, and make those cells become even more specialized. Okay. And what about a sponge? It's a legitimate question. Well, in the case of a sponge, you get something that's very similar to gastrulation, at least in some sponges. I can't speak for all the sponges, but if you sort of visualize a blastula-like cluster of cells, which is a sponge embryo, basically a sponge egg will undergo all these cleavage divisions going from zygote to 2-cell to 4-cell to 8-cell, all the way until it gets to something like a cluster of cells. And I think they might even call them pseudoblastulae. But the pseudoblastule basically settles out in the bottom and it forms a little indentation that's at the top, which then becomes the spongiocele. So in other words, when we get from this stage we have right here, over to something that looks legitimately like a sponge, like the ones we saw in the video, right? It really is just a matter of modification of existing structures. The one difference is that in the sponge, we don't find the two germ layers. We don't see endoderm and ectoderm becoming distinct from each other. We just see one kind of generic bunch of cells that are all going to become part of the sponge. Yeah, certainly a sponge has differentiation, but the way in which the cells are specialized follows a little bit of a different pathway. It turns out that those things we call the amoebocytes, those are the ones that are undifferentiated, and they ultimately give rise to the other kinds of cell types. But basically, one thing that you could say that is true for both sponges as well as sea anemones is the fact that they go through these stages of development where they initially form a cluster of cells and at some point they actually form a little indentation, whether that's an actual blastopore or this little indentation, the future spongiocele at the top. It's kind of like an academic question. The basic process of a zygote proliferating into a generic cluster of cells and then undergoing some type of morphogenesis to create a structure with a whole that is held in common by all of animals. And it's been elaborated upon by the rest of the animals, but basically at some point in our distant past, we are really nothing more than a blob of cells that created a little hole. And so now getting back to our original phylogeny, and I'm going to go ahead and get rid of this because we don't need him anymore. We can basically say that in addition to multicellularity right here, there, what we have also is the origin of morphogenesis slash something like gastrulation. The other thing you might have taken note of is that we compared two animals. One was a sponge, the other one was a cnidarian. The sea anemone was a cnidarian, and the cnidarian was actually different from the sponge in actually having two germ layers. Eumetazoa would be characterized by having germ layers, specifically two germ layers, endoderm and ectoderm, which basically means that they have true tissues. And that's a characteristic of the Eumetazoa. In other words, it's a characteristic that had to have evolved somewhere along the lineage. The Porifera did not get that, but the Eumetazoa did. And the presence of true tissues is something that is held along that entire lineage. We have true tissues as well. Basically, we are bilaterians. Radiatins also have true tissues. One difference is that bilateria will have three germ layers. The other thing to note about the eumetazoans is that we do our digestion in a completely different way. Digestion, in the case of things like sponges, is largely intracellular, which is a characteristic that is shared with the quantiflagellates. Basically, you could say, well, the ancestral state, we had intracellular digestion. That means that the food particles were taken into a food vacuole and then digested inside of the cell. Whereas in the case of the eumetazoans, like that sea anemone, the sea anemone would do things extracellularly. Sea anemone kind of looks like this. It's the one with the tentacles around the outside. The food particles will go inside the animal's stomach, inside of the gastrovascular cavity. And digestive enzymes are then secreted into the gastrovascular cavity, and the food then is kind of digested and then absorbed into the walls of the gastrodermis. And so that's a completely different thing. Basically, our digestive tract works on the same basis. We digest things in the lumen of our GI tract, and that characteristic is also there as kind of like a synapomorphy of the eumetazoa that separates us from even the more basal of our animal relatives. The fact that we have extracellular digestion, and that's a characteristic of the eumetazoa. Now the split that occurs between the radiata and the bilateria, the split there is largely a distinction between two germ layers versus three. Two germ layers, we have a term for that, we call that diploblastic. Whereas in the case of three germ layers, we say that is a triploblastic body plan. And there's more to it than that. Triploblastic means we've got a third germ layer. In addition to endoderm and ectoderm, we also have the plus mesoderm. And mesoderm is the germ layer that we associate with muscle, muscle being a dedicated contractile tissue. As vertebrates, our bone and our cardiovascular system, including all the blood, is also derived from mesoderm. So mesoderm is a germ layer that is associated with muscle and a whole bunch of other characteristic cell types, tissue types that we have in more complex animals that are associated with movement. Endoderm and ectoderm, for all their glories, they're not especially good at contracting, whereas mesoderm is something whose whole reason for being is to contract and allow the animal to move around. Okay, so we're going to be talking about that next.